Every thought, every muscle contraction, every heartbeat begins with a tiny electrical signal. But how does a nerve cell generate electricity? How does the membrane suddenly change from negative to positive and then reset again? In this video, we'll break down the action potential step by step, clearly, logically, and in a way that actually makes sense for exams. First, we'll understand resting membrane potential. Then we'll move to threshold and depolarization. After that, we'll analyze repolarization and hyperpolarization. Then we'll discuss refractory periods, very important for exams. And finally, we'll connect everything to clinical applications. At rest, the neuron is negatively charged inside relative to outside. The resting membrane potential is about minus 70 millivolts. Why negative? Because potassium concentration is high inside the cell and sodium is high outside. Potassium leaks out through leak channels. The sodium-potassium pump maintains the gradient by pumping 3 sodium ions out and 2 potassium ions in. This creates and maintains the negative resting potential. If the membrane is stimulated and depolarizes to around minus 55 millivolts, threshold is reached. At this point, voltage-gated sodium channels open rapidly. This triggers a massive sodium influx. The action potential follows an all-or-none principle. If threshold is not reached, no action potential. If threshold is reached, full action potential occurs. Once sodium channels open, sodium rushes into the cell due to both electrical and concentration gradients. The membrane potential rapidly rises. It crosses zero and becomes positive. At the peak, it reaches around plus 30 millivolts. This phase is called depolarization. It is extremely fast. Sodium channels quickly inactivate. At the same time, voltage-gated potassium channels open. Potassium moves out of the cell. The membrane potential returns toward negative values. This phase is called repolarization. Potassium channels close slowly, so more potassium leaves than necessary. The membrane becomes even more negative than the resting potential. This is hyperpolarization. Eventually, potassium channels close, and the membrane stabilizes again at minus 70 millivolts. During the absolute refractory period, sodium channels are inactivated. No matter how strong the stimulus, another action potential cannot occur. During the relative refractory period, some sodium channels have recovered. A stronger than normal stimulus is required. These refractory periods ensure action potentials travel in one direction and prevent overlap. Local anesthetics like lidocaine block voltage-gated sodium channels. Without sodium influx, action potentials cannot propagate. Hyperkalemia reduces the resting membrane potential, affecting excitability. Myelination increases conduction velocity through saltatory conduction. Understanding action potential makes neurophysiology and pharmacology much easier. Let's summarize. At rest, the membrane is negative. Reaching threshold opens sodium channels. Sodium influx causes depolarization. Potassium efflux causes repolarization. Refractory periods ensure controlled, one-way conduction. If you understand this sequence, action potential becomes logical, not memorized. If this explanation made action potential finally clear, make sure to subscribe. We're building physiology step by step, clean, exam-focused, and easy to remember. Next, we'll simplify synaptic